everyone. I am Dr. C. S. Kulkarni, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with beta oxidation steps. In today's session, we will start with further cycles in fatty acid metabolism. Then we will have energetics of beta oxidation. Then we have defects in beta oxidation. Then we have fate of propionyl CoA, and lastly we have alpha oxidation. Good morning everyone. Let us begin the topic. Further cycles. The newly formed fatty acyl CoA will sequentially undergo further cycles of step 1, 2, 3 and 4 of beta oxidation until the fatty acid is completely converted to acetyl CoA. Coming to summary of beta oxidation. When one molecule of palmitate undergoes beta oxidation, the next reaction is written as palmityl CoA converted to 8 acetyl CoA plus 7 FAD is converted to plus 7 FADH2 plus 7 NAD plus is converted into plus 7 NADH plus 7 H2O gives plus 7H plus, and lastly plus 7 CoA SH is added to the palmitate. This is a diagram showing beta oxidation of palmitic acid. The palmitic acid is broken down into 8 molecules of acetyl CoA. Coming to energetics of beta oxidation. Palmitic acid needs 7 cycles of beta oxidation, so it gives rise to 8 molecules of acetyl CoA. Every molecule of acetyl CoA, when oxidized in TCA cycle, it gives 10 molecules of ATP. Each molecule of FADH2 produces 1.5 molecules of ATP, and NADH generates 2.5 molecules of ATP when oxidized in the electron transport chain. Hence, the energy yield of from one molecule of palmitate may be calculated as 8 acetyl CoA into 10 is equal to 80 ATP. 7 FADH2 into 1.5 gives 10.5 ATP. 7 NADH into 2.5 gives 17.5 ATP. The gross total is 108 ATP, and the net yield is 108 minus 2 is equal to 106 ATP. The initial activation reaction, the equivalents of two high energy bonds are utilized. The efficiency of beta oxidation is about 33%. Coming to regulation of beta oxidation. The availability of free fatty acids regulates the net utilization through beta oxidation. The level of free fatty acids in turn is controlled by glucagon to is to insulin ratio. The glucagon increases free fatty acid level, whereas insulin decreases the free fatty acid level. CAT1 is the regulator of entry of fatty acid into mitochondria. Melanin CoA decreases CAT1 activity. Thus, during de novo synthesis of fatty acids, the beta oxidation is decreased. Coming to defects in beta oxidation. Abnormalities in transport of fatty acids into mitochondria and defects in oxidation can lead to deficient energy production and oxidation of long chain fatty acids. Common features are hypoketotic hypoglycemia, hyperammonemia, skeletal muscle weakness, and liver diseases. Acyl carnitine accumulates when the transferase or translocase is absent. Dietary supplementation of carnitine has been found to improve the symptoms in some cases. Coming to organic acidurias, they are disorders of metabolism of fatty acids, branch chain fatty acids, then aromatic amino acids, citric acid cycle. The incidence of medium chain acyl CoA dehydrogenase deficiency is about 1 in 2500 live births and is the second most common error in inborn error in metabolism. They are all characterized by the accumulation of organic acids in bodies and in body tissues and their excretion in urine. The patients present with acidosis, vomiting, convulsions, and coma. The children often die in infancy. In case they survive, there is severe mental and physical retardation. Diagnosis is confirmed by showing the presence of organic acids in urine by chromatography. Dietary restriction, cofactor therapy, and substrate removal are the general lines of management. Here are some important organic acidureas. Methyl malonyl aciduria. Its deficient enzyme is methyl malonyl CoA mutase or B12 coenzyme. Its clinical features are ketoacidosis, hypotonia, hypoglycemia, hyperammonemia, and hyperuricemia. Propionic acidemia. Its deficient enzyme is propionyl CoA carboxylase. Its clinical features are ketoacidosis, hypotonia, vomiting, and lethargy. MCADH deficiency. Its deficient enzyme is medium chain acyl CoA dehydrogenase. Its clinical features are acidosis, hyperammonemia, hypoglycemia, and fatty liver. LCADH deficiency. Long chain acyl CoA dehydrogenase is the deficient enzyme. Its clinical features are non-ketotic hypoglycemia, low carnitine, increased acyl carnitine, and yeah. Then glutaric acid. Its deficient enzyme is glutaryl CoA dehydrogenase. Its clinical features are ketoacidosis, convulsions, progressive neurological defects, and cerebral palsy. The odd chain fatty acids are oxidized exactly in the same manner as even chain fatty acids. However, after successive removal of 2 carbon units, at the end, one 3 carbon unit propionyl CoA is produced. Coming to fate of propionyl CoA. First enzyme to act on it is carboxylase. Propionyl CoA converted to D-methyl malonyl CoA by a biotin-dependent carboxylase. Biotin is a member of vitamin B complex group. One molecule of ATP utilized is to supply energy. Then the next enzyme to act is racemase. The racemase acts upon D-methyl CoA to form L-methyl CoA. Here you can see the metabolism of propionyl CoA. Propionyl CoA plus CO2 is converted into D-methyl CoA in the presence of enzyme propionyl CoA carboxylase, which is biotin dependent, and ATP is converted into ADP. Then this D-methyl CoA is converted into L-methyl CoA in the presence of enzyme racemase. Then L-methyl CoA is converted into succinyl CoA with the help of enzyme mutase. Third enzyme to act is mutase. The L-methyl CoA is converted into succinyl CoA by L-methyl CoA mutase. The reaction needs vitamin B12 coenzyme. The succinyl CoA enters reset cycle, finally converted to oxaloacetate and is used for gluconeogenesis. Propionyl CoA is also derived from the metabolism of valine and isoleucine. The next point is propionate is gluconeogenic. Ordinary fatty acids are cleaved into acetyl CoA, which enters into the Krebs cycle and are completely oxidized to CO2, and hence, as a general rule, fatty acids cannot be used as for gluconeogenesis. However, propionate is entering into the citric acid cycle at a point after the CO2 elimination steps. So propionate can be channeled into gluconeogenesis. Thus, three carbon atoms, carbon units from odd chain fatty acids are gluconeogenic. Cow's milk contains significant quantity of odd chain fatty acids. Coming to inborn errors of propionate metabolism. Propionate carboxylase deficiency. It is characterized by propionic acid acidemia, ketoacidosis, and developmental abnormalities. Methylmalonic aciduria. Some patients respond to treatment with pharmacological doses of B12. This group will have defective formation of adenosyl beta 12 with deficient mutase activity. The second type will not respond to cyanocobalamin and has deficiency of the enzyme racemase or mutase. The methylmalonate affects the metabolism of brain, leading to mental retardation in these cases. Coming to alpha oxidation, it is a process by which fatty acids are oxidized by removing carbon atoms one at a time from the carboxyl end. The process is important in brain. The fatty acids does not need activation. Hydroxylation occurs at the alpha carbon atom. It is then oxidized to alpha ketoacid. The ketoacid then undergoes decarboxylation, yielding molecule of CO2 and a fatty acid with a one carbon atom less. This process occurs in the endoplasmic reticulum, where does not which does not require CoA and does not generate energy. Some fatty acids undergo alpha hydroxylation in peroxisomes also. Alpha oxidation, mainly used for fatty acids, have a methyl group at a beta carbon and with which blocks beta oxidation. A major dietary methylated fatty acid is phytanic acid. It is derived from phytol present in chlorophyll, milk, and animal fats. Here is a diagram showing straight chain fatty phytanic acid. Here ends our session. Kindly like, share, and subscribe the channel. Thank you.